Welcome to Matri Academy. Let's begin the chapter 22 DNA sequencing. The first biological significant nucleic acid to be sequenced was that of East alanine by Robert Hawley in 1964. It is having 76 nucleotides. In 1976, the entire 3569 nucleotide genome of the bacteriophage MS2 was sequenced by Walter Fires. Dideoxy method, chain termination method, enzymatic method, or Sanger and Coulson's method of DNA sequencing. These all are the name of dideoxy method of DNA sequencing. Chain termination method of DNA sequencing was invented by F. Sanger and Coulson in 1977. Question asked in ARS 2007. Used to sequence 800 base pair long DNA fragments, while chemical method of DNA sequencing is used for smaller DNA fragments. Page permits sequencing of 400 to 500 base pair DNA fragments from a single gel. Question asked in ARS 1998. The dideoxy NTPs do not have 3' hydroxyl groups. Clinue fragment is used in dideoxy Sanger's method of DNA sequencing. At present, the sequence of a maximum of 700 to 800 bases can be determined using one strand of DNA as a template. Since the two strands are sequenced from the two opposite ends of the DNA duplex, DNA molecules up to 1.5 kb can be sequenced using one pair of primers by chain termination method of DNA sequencing. The next one is the chemical method, Maxam and Gilbert method of DNA sequencing. These both are the of chemical method. In a chemical method of DNA sequencing, DNA segments are end labeled either at 5' or 3' end. Question asked in ARS 2007. The range of chemical method of DNA sequencing is less than that of Sanger's method. Sanger's method is more in use as compared to chemical method of DNA sequencing. With current knowledge, it is possible to record the order of 400 bases from the autoradiogram of a sequencing gel. Next one is the high throughput genome sequencing. These are the two methods. Whole genome shotgun method used in the sequencing of prokaryotic genome which are less repetitive. Hierarchical sequencing method, also known as clone by clone approach, require construction of genetic linkage map of an organism. NSG technology, next generation sequencing technology. Question asked in ARS 2012. Use 3 platform for sequencing. 454 pyro sequencing platform in which 400 to 500 nucleotides can be sequenced at a stretch. Solexa 100 nucleotides can be sequenced at a stretch, and solid 50 nucleotides can be sequenced at a stretch. So guys, these all are the rapid notes of DNA sequencing and various questions, important questions which are going to ask in the next coming examination of ARS NET, IARI and PhD, SRF JRF. So let's tune to my channel. Always like and subscribe.